breaks. We do have, yes, see, I told you it was a team effort. We have evaluations on very bright colored paper today. And so if you would take time before you leave to either fill them and leave them on the table or put them out by the front, I really, really, really appreciate it. And we read and hang on all of the comments and feedback that we have. And so I would like to introduce Martha, who will kind of get us going program-wise. And that's it for now. Martha?
Thank you, everyone. I have the pleasure of introducing our first speaker, Dr. Martin Tenniswood, and I was thinking about this in the context of Dr. Tenniswood and then kind of all of this. Eileen mentioned an expression, and I had another one, which was something parents say to kids. They say, "You are known by the company you keep." We've all heard that. And I was thinking about this last night, and I said to my husband, you know, when I think about the totality of an event like this, and I think about, as Eileen said, this is, for True Life, this is a huge team effort. It's huge. It is, above all, it is a team thing. And then when I think about this group and the people that I've met over the last 4 years that I've been here and the work that's done and the lives that women dealing with breast cancer are living, it's really remarkable. And then in particular, I was thinking about Martin Tenniswood, who we as True Life feel very privileged to be associated with. And I'll tell you a little bit about him. Just from a bio perspective. Martin Tenniswood was born in Uganda and lived in England, Newfoundland, Rhodesia, Scotland, and Ontario before he finished high school. He received his BS degree in chemistry from Trent University in, I'm leaving out all the dates, I hope you're good with that. From Trent University in Peterborough, Ontario, Canada, and his PhD in biochemistry from Queen's University in Kingston, Ontario, Canada. Since then, it's been research and studies and travel and publishing and so forth until 2008, when Dr. Tenniswood came, it says, back to New York State as an Empire Innovations Professor at the Cancer Research Center in the School of Public Health, University of Albany, and he became director of the center in 2009 and has been there since. He's spoken for True Life a number of times on topics related to cancer, better understanding cancer, the biochemistry of cancer, and much more of what he'll speak about today. But what I want to say in a very heartfelt way to him from True Life is that we feel very honored, as Eileen mentioned, that he would want to spend time with us, to present to us, and meaning all of us, not just True Life, but the fact that this is, as I always say to presenters, this is a smart, savvy group who really get it and really understand the complexity. So thank you so much, Dr. Tenniswood.
So, I don't want to use the microphone if I don't have to. Can you guys all hear me? It's a problem, you stick up your chest and tell me. If you can't hear me, you can't put up your hand because you can hear what I just asked. So, are you okay with it like this? No. You want to do the microphone? I'm going to take it with me. But you see, the problem is, I've got two hands, so my brain only handles one hand at a time. So, if I drop off, let me know. So first of all, I'd like to thank True Life for inviting me back, because that means I didn't really screw up the last time I was on. So, the other thing I want to tell you is I'm going to talk to you about some biochemistry and some molecular biology. I'm going to do it in the simplest way I can, because it's a very complicated field, and I don't want to dumb down the science, but I also think it's very important for you to start to understand some of the complexities that scientists are dealing with in terms of what breast cancer is. I have given a previous talk at True Life talking about the fact that cancer, breast cancer in particular, is a lot of different diseases. It's not a single disease. And in fact, I would go as far as saying that since every individual that gets breast cancer is an individual, we need personalized medicine as the way to approach the disease. We haven't got there yet. But what I'm going to show you on the way is where we have got to, and the kinds of advice that you're going to hear in the next lecture, how it actually applies to each of the different kinds of cancer, of breast cancer. So this is where I actually work and hang out. It's actually a marvelous photograph that's been taken that even manages to make the, as I said, I'm only one-handed. This actually shows the Cancer Research Center. It's in Rensselaer, not in Albany. So I'm on the other side of the river. But one of the marvelous things about good photography is that you actually make the loading docks look very sexy. So it's not quite as fancy a building as it looks, but it is still pretty spectacular. Not from the point of view of the building and the edifice, but the people that we work with who are in there. So what I want to do today is to talk to you about a number of different issues. Some of this will be old hat to many of you, but I want to bring it down at the end and talk about the fact that what we now understand about breast cancer and the types of molecular subtypes that we've got actually helps us with figuring out how to live with that particular kind of cancer that we've got. And how that's really where there needs to be the next revolution in terms of patient care for survivors. So I'm going to tell you, and I will, for those of you who want to keep count, there's 22 slides left in my deck. I'm going to talk to you very briefly about basic breast cancer statistics because that's important from the point of view of how well we're doing. It also tells us how bad we were doing. I'm going to talk about the good stuff. I'm not going to terrify anybody, I promise. I want to point out to you and explain to you that breast cancer is in fact many diseases. And I want to talk to you about the fact that the histopathology that most of you know about your disease tells you something about the type of breast cancer, but it doesn't really inform you at the really advanced level anymore as to how to treat the breast cancer. And I'll show you why in a few minutes. But basically, we're now doing molecular analyses of breast cancers to determine this, what's called the subtype of the cancer, and so that we can actually dictate treatment options. Okay? When I started in this business, and I'm very glad that the dates were left out, it only reminds me that I've been doing this for far too many years in some respects. But back when I started doing, I was one of the first people to develop the estrogen receptor binding assay that's used to identify estrogen receptors. That paper came out in 1975. Okay, so that tells you, gives you an idea that I've been around a long time. But basically, that was the very first molecular subtyping that could be done. Did the breast cancer have estrogen receptors or did it not? Okay? At the time, that really didn't help with the treatment options because the treatment options at the time were radical mastectomy for everybody. Okay? And the surgeons that I worked with wanted to be able to not do radicals, but MDs, usually surgeons, have to work on the side of caution, so they'd operate on everybody as though they had a single disease. It's clear now that's not the case. Okay? The other thing I want to do when I get to the end of this is to talk to you a little bit to lead in to the next talk. The fact that understanding the molecular changes that occur in breast cancer actually provides everybody in this room who is a survivor, long-term or short-term survivor, or somebody still in treatment, it gives you all kinds of options for personal action. And basically, it's understanding these things that become critically important for wellness, not just for living with the disease, but from genuine wellness. Okay? So, the quick statistics, and these are actually kind of standard. As a molecular biologist trying to get grants from the NCI, the National Cancer Institute, the first thing I've got to do is take the statistics. So it's inbred in any molecular biologist doing research. Last year, there were just over 230,000 new cases of breast cancers diagnosed in the United States. And it was the second leading cause of death in the the States in terms of cancer-related deaths in women, and that was about 40,000. Now, there's bad news in this, that all of us who are working hard to develop new cures focus on, and that's that we're failing at least 40,000 women a year. There's no doubt about that. We're working hard to change that. But there's also very good news in this. There's almost 200,000 women who are going to live 10 plus, 15 plus, and 20 plus years on. The number of women in here that are now long-term survivors, I'm actually impressed by the number of people that have come today who are long-term survivors. It's very important. The key thing with all of this is 1 out of 8 women, that's 12% of all women, will actually be diagnosed with an invasive form of breast cancer. And what we have been looking at for years now is how to improve the survival and treatment of patients with invasive breast cancer. So when does it occur and how is this informative? Well, if you look at the diagnosis of breast cancer, look at the risk factors, there are 2 major risk factors. And you can't avoid either of them, I'm afraid. First of all, it's gender. Women have much higher rates of breast cancer than men. There is actually 2 lines on this curve, a pink one for women, and there's a blue one for men. They're very, very heads off of the axis. So there are very few cases of male breast cancer. They're almost all the result of a hereditary breast cancer gene being inherited by the man. And these men have a very high propensity of getting prostate cancer. So they don't get away scot-free. Men do not get away scot-free. I have to say that because otherwise I won't really feeling that I've not done my other half of my job, which is prostate cancer research. So basically, if you look at the rate of increase of breast cancer in women and look at the ages at which women are screened for breast cancer, it's mainly between the ages of 50 and 74. This is the major peak area. Women are in that areas, as you are probably all very well aware, are perimenopausal or postmenopausal in terms of their diagnosis. There are patients who get breast cancer much earlier. They exist. And their prognosis and treatment should be different than it is for than it is for women who are postmenopausal. And I'll show you how that works in a few minutes. And the key feature of this is that 85% of breast cancers occur in women with no family history of disease. They are the result of random genetic changes in the woman's genome in the breast tissue itself, not in the rest of the body. Okay? If it's in the rest of the body, if it's occurring in other tissues, then that's going to be a hereditary form of the disease. The thing is that no matter what we do in terms of treatments and understanding the disease, understanding that there will always be, in the future, women will still get random changes in breast cancer, in the genome of their breast tissue, and they will still be exposed and likely to get the disease, no matter what we do. This is just something that's going to be around. What that actually says is that the most important thing we can do long-term is to screen patients and then have early diagnosis and then have very good therapies for patients who are diagnosed early. We don't do either of those very well as a scientific society yet. We're actually getting there. The good news then in all of this is that there are 3.1 million breast cancer survivors as of January 2017, which is the last date that the data is actually available for. 400,000 women are currently being treated for some form or other of breast cancer. 1 million women have finished their therapy, and there's now 1.5 million women who are now long-term survivors. That's a huge change and a huge improvement over what we were seeing when I started work, where the average mean survival of 5 years was less than 50%. So there's been a huge improvement. We're nowhere there yet all the way through. It can't get much better than that. And I mentioned to you there are additional risks for breast cancer. One of them are genetic risk factors. They're the major one. 5 to 10% of breast cancers are linked to specific gene mutations. And the common ones that you've all heard about are BRCA1 and BRCA2. Therefore, all of these cancers are referred to as hereditary breast cancers. And you should be aware that a woman's risk of breast cancer doubles if she has a first-degree relative, a mother, aunt, sister, or daughter, who's been diagnosed with breast cancer. That doesn't mean it's hereditary, but it means that there's could also be environmental factors that are influencing the development of breast cancer. And one of the things that we really need to work on as a society are what I refer to as the modifiable risk factors. There's no doubt that smoking increases the risk of breast cancer. And we can go through all the molecular biology of that. And it's because the carcinogens in cigarette smoke actually land up getting into the circulation of the smoker and are transited around to target organs where they're actually metabolized to become very active DNA damaging agents. Smoking is devastating for a whole bunch of different diseases, cancers. Obesity and type 2 diabetes, which are actually foisted on us in many ways by the entire food environment that we live in, which I think is critical. These are actually high-level risk factors for prostate cancer, bladder cancer, breast cancer, and a number of other cancers. And we always list exposure to environmental carcinogens and exposure to xenoestrogens such as endocrine disruptors that you hear a lot about in the literature. But these probably constitute a very small proportion of the patients that actually are diagnosed with breast cancer. That's directly from exposure. Okay. I want to talk to you very briefly about the anatomy of the human breast, and the reason I'm doing this is so that you understand that what happens in breast cancer is just a disruption of what the breast is normally expected to do in life. Okay. So if you look at the physiology, the breast consists of a large amount of adipose tissue, and the key features of this are the ductal structures. And the ductal structures are designed for use, actually predominantly active during pregnancy. The epithelial cells of the mammary gland, particularly the lobules, which are these little bundles, grape-like bundles at the back end of the duct, are where milk is produced. And milk is produced in these tissues during pregnancy as a result of the presence of prolactin and estrogen. Okay. And what happens during this phase is when you become pregnant in these tissues, they're very small to start with, and anybody that's had kids knows this. The breasts enlarge. It's because these tissues, in response to estrogen, actually divide and proliferate, so you've got many more cells making milk for the child. And there's two regions. This is the lobules, and the region towards the nipple is the duct. Okay? I'm showing you this because, in fact, this is what a duct looks like. This is the bit in the middle is known as the lumen. We've got the epithelial cells that actually are making and transporting the milk. And the myoepithelial cells are a very thin layer of muscle-like cells around the outside of the duct. And they're the muscles that contract in response to oxytocin, which is the letdown that you see when you see your child. You start to lactate. Okay? The important thing about this is after lactation, what is supposed to happen, and what happens in fact in healthy women, is the lactation of the cells undergo apoptosis and they all die. Okay? So the excess of the tissues is removed as a result of what's called active cell death. So the mammary gland gets larger in response to estrogens, and when the estrogen stimulus is removed, the cells normally die. Okay? And what has happened, and what happens during this entire process, is that the mammary gland, in fact, is primed to respond to estradiol. And what it does is it initiates DNA replication and proliferation. So what it's doing is it's making more mammary gland cells. Afterwards, those cells should die. Okay? What happens in the case of a breast cancer, not as a result of pregnancy, but somewhere during their lifetime, they receive a series of mutations I'll talk about them in a few minutes. And the end result of those mutations is that they no longer respond properly to estrogen, And more importantly, they no longer respond to the withdrawal of estrogen. Okay? And what happens is instead of dying, they survive. So you land up with damaged cells that survive. What causes that initial damage really isn't fully understood. We know we can track all kinds of pathways in the cell, but really we don't know what causes the initial mutations that land up causing these cells not to respond properly. And if you look at histologically at these and you give this tissue samples from a patient with breast cancer to a pathologist, they have figured out 15 different kinds of breast cancer. Okay? It's enormously complicated. But you can look at these and they've given them all kinds of different names. I'm not worried about the names. What I'm concerned about are two things. One of which is the 5-year relative survival risk. And you can see that if you've got a tubular adenocarcinoma, it's actually a benign disease in essence. 100% survival. And that's after surgery to remove the tumor, but it's a very benign disease in essence. It doesn't metastasize. And if you look at this, and look at the 257,000 cases, this was actually taken, I think, 3 years ago, from the table from results from 3 years ago. And you look at it, the most predominant disease is invasive ductal carcinoma. 183,000 individuals got the same pathological kind of disease. Okay? Lobular carcinoma is about another 20,000, so that's 200,000 out of 257,000 cases have come from two kinds of cancer. Okay? Cancer from the point of view of the histology. And the survival rates, the 5-year relative survival rates for all of these, right down to serous adenocarcinoma, which is 81%, is actually not bad. It's not as good as it should be, but it's not bad. What you land up with, though, is a couple of diseases like inflammatory carcinoma, which has a 34.1% 5-year relative survival. Okay? So we're doing very poorly on this particular breast cancer, this histological form of breast cancer. So if you look at this from the point of view of a pathologist, there are different kinds of disease. This is ductal carcinoma. This is the one that's 180,000 plus cases a year. And if you look at what ductal carcinoma looks like, instead of having a single ring of epithelial cells, what in fact you have is a infilling of the lumen of the tissue. You have a large number of cells here, and you have what's called a cribriform structure. Okay? Cribriform, this is actually a section through a cribriform, a DCIS. This cribriform structure is known as ductal carcinoma in situ. I suspect some of you have had that as a diagnosis. Okay? And this is in fact got a very high survival rate, close to 100%. Okay, with treatment. Invasive cells, when you have a tumor cell where the tumor has filled in the entire lumen, literally physical pressure breaks down the lumen and allows the cells to escape through the myoepithelial lining of the tissue. And that leads to invasive cells here. Those invasive cells are the ones that pathologists and therapists have to be very careful about. Those are the ones that can lead to metastasis. So if you see invasive ductal carcinoma, we know that needs to be treated. It needs to be treated to prevent metastatic progression. So if you look at what we call the precursor lesions, and I'm going to point out to you in a couple of minutes that these really aren't precursors necessarily. Okay? What you see is a normal duct. You get ductal hyperplasia. This is a benign disease where you can start to see extra cells filling into the ducts. The atypical ductal hyperplasia fills out a bit more. And still, you've got different cells. You can start to see breakdown of the structure of the cells. You see DCIS can form, and DCIS with microinvasion, where it's just starting to creep through, forms. And then you've got invasive, where you've got lots of stuff breaking through. Those are what the pathologist will report on. trying to figure out what you do to treat those different diseases is complicated. And for many, many years, people treated any DCIS was treated as though it was actually a precursor lesion. Okay? We know now that not all of the DCIS is a precursor lesions. And if it was a precursor lesion, you actually had what is now a lumpectomy or modified radical mastectomy, depending on where you are. And basically, this was classified then as a cure. Okay? But one scary piece of news with this is related to dissemination. Okay? And I want to point out to you that the way cells, once they've got out through the myoepithelial layer and they're embedded into the other tissues, what happens is they metastasize through the lymphatic and circulatory systems. Okay? This is the lymphatic system. This is a separate system from the circulation, general circulation. It's actually designed to take fluids that are being taken into tissues and push them back into the circulation. And in fact, the way these work from wherever they are, in the lymph nodes down the legs, everything's pushed very slowly. It's not pumped, it's just pushed back through the lymphatics, and everything's released at the subclavicular vein. Okay? So that basically it's all pushed through slowly. And the reason this is important, there's two reasons it's important, is because breast cancer is actually metastasized predominantly through the lymphatics. And what happens is they get into the lymphatic system and move and then released into the circulation. When they get into the circulation, they can actually travel to distant organs, and they'll colonize, first off, they'll colonize the lymph nodes, the bone, the liver, the lungs, and the brain. That's specifically what we as molecular biologists and drug developers are trying to stop. The sad thing about this in many ways, and this was actually a hypothesis that was first put forward by Stephen Paget in 1889 in a publication, and we're still working on this whole process. The reason I want to point this out to you is because in a couple of minutes I'm going to talk about sentinel lymph nodes. The drainage system for cancer, okay, the cancer cells out of the breast, is into the lymphatics, and they then go, as I said, back to the circulation. So one of the things that you will see when you're diagnosed and you have a surgery, they'll take sentinel lymph nodes to figure out whether they are involved. What that means is they look in the lymph node and count the number of tumor cells that might be in the lymph node. The ones that are in the lymph node are not a good sign for metastatic regression. And basically what it is, it's a number that tells you how many lymph nodes have been involved and how many cells there are in each of the lymph nodes. Okay? That's one of the keys to figuring out whether or not we need to have a very major intervention, or whether or not we can have a less rigorous and less robust intervention. Okay? The decision between those is actually entirely a question of lifestyle and life choices in many cases. I don't think doing a really robust intervention for many of these diseases is necessary. I don't think it changes lifestyle and life expectancy. What it does do is it causes a great change in the quality of life. So it's a very complicated equation that every single one of the survivors has to figure out for themselves. Okay? I can say this all, and I don't have a problem. So I have no problem. As last check, I did not have male breast cancer. So if you look at the different ways we analyze disease, okay, one of the important things is you can go through the different histologies and you can determine whether you've got lobular carcinoma, ductal carcinoma, infiltrating carcinoma, or inflammatory breast cancer. Those histologies will tell you a little bit based on other people that have had the disease, on the epidemiology of the disease. They'll tell you whether you have, they'll tell you about the tumor itself, but really it doesn't usually help in the choice of treatments. Okay? It tells you a little bit whether it's a very, very serious disease or not. And what in fact turns out to be really important is the diagnostic that the surgeons are using, the TNN classification. They talk about the size of the tumor, the nodes, the number of the lymph nodes that are involved, and whether or not there's any evidence on MRI or other technologies of metastatic progression. And understanding the tumor helps to define kind of interventions needed. You can have a very large tumor, but if there's no lymph nodes involved and no evidence of metastasis, that's one that you can probably treat very, very conservatively and watch. If you have a small tumor, but there's tons of mets, then you have a different disease. And this is one of my key points, that everybody has a different disease. Not just as a group, but all of you individually essentially have a different disease. And that's becoming more and more evident when we look at different molecular subtypes. So we now break down cancers, breast cancers, into a variety of different subtypes. The three that I'm going to talk about briefly are ER/PR positive tumors. They have a phenomenally good response rate, and they have a very good survival rate. HER2 tumors, ones that express a protein called BRB2, and I'll talk to you about that in a few minutes, actually have a good prognostic indication. And if you have triple-negative breast cancers, in other words, triple negative is they don't express ER, they don't express progesterone receptor, and they don't express HER2. They classify every other tumor is lumped in as triple negative. Okay? But these can now be used very successfully to individually tailor treatments. Okay? So I want to spend a few minutes explaining to you some of the treatment therapies, and because, as was mentioned, I think one of the things that you'll see out in chemist, you'll see evidence of that in a couple of minutes. If you look at this, there's a bunch of lessons from breast cancer screening. First of all, early diagnosis is critical to overall survival. And we're getting into all kinds of arguments about mammography, about self-exams, and whether or not they're worth doing. I can't imagine a society that does what they've done with prostate, by the way, and said the PSA is useless. And now the diagnosis for prostate cancer is going back to a digital rectal exam that was done in the 1950s. They've actually abrogated responsibility to develop the new tests, molecular tests. Okay. They're coming, but there's going to be a gap of about 10 years when we're going to have an increase in the diagnosis of later stage prostate cancer because there's no diagnostic technique available. So for breast cancer, I think it's going to be critical to keep doing what we're doing until somebody comes up with a definitive diagnostic test that hopefully is non-invasive. And one of the things that's obvious, I think, is that survival rates decrease a bit with lymph node involvement, especially with advanced lymph node involvement. And if you've got evidence of metastasis, that's actually a poor prognostic indicator, although treatment for metastatic disease has actually improved very greatly recently for breast cancer. Now, one of the downsides of this is that the emphasis on early detection has led to an increase in the number of individuals who are diagnosed with ductal carcinoma in situ. Last year, there were 60,000 diagnoses of DCIS. And it turns out from the literature and looking at historical studies, only 40% of these patients ever progress to invasive disease. So the precursor lesions I talked about, in the case of DCIS, a lot of them just disappear. And it depends on when you catch them. And there's a scientist in Canada called Stephen Nayrod who's been studying this now for a number of years. And it turns out that you get little growths like this in the breast quite often. And they grow to a certain size, just big enough to be diagnosed. And if you follow them, they'll disappear. Okay? And that's one of the problems. Because basically right now, we have no way of distinguishing the DCIS that's going to progress and the DCIS that's going to disappear. And so they're all being treated as though they're precursor lesions. That's not good. It's good for everybody that they're getting treated, but it's not good because it actually means that we're over-treating a significant population of women. Okay? And we need better technologies for diagnosis. That's a different picture and a different diagram on my part. But if you look at basically what we're trying to do with cancer therapies at the moment, we have an average life expectancy of women in New York State is 80.4 years. It varies depending on their ethnicity. It's lower for African Americans for a number of reasons. It's higher for Asian Americans. The average age is 84. The average age for diagnosis, okay, is 67 for women with breast cancer. So really, in many ways, what scientists are trying to do is buy enough time for everybody that's diagnosed with breast cancer to die of something else. That's really what we're doing. Nobody's pretending that we're going to get you correct. And obviously there are women in the room, and a lot of women in the state, and in the States in general, who actually have been diagnosed with breast cancer much earlier. Okay? So our job for those is not an average of 13 to 14 years, it may be 20 to 30 to 40 years. And we have to work on all of those. The shorter difference and the much longer differences. They are different diseases, without a doubt. Okay? So our immediate goal is to provide 14 years of healthy life. The way you do that is to prevent metastatic progression. And essentially what we're doing for most women who have breast cancer is we're turning it into a chronic condition. Okay, that's really what most of these therapies do. And what we really want to do is just cure it. But that's not really what we're doing right now. It's what we're trying to do. And basically, what we also need to do is ensure a high quality of life and minimize treatment side effects. And those become a major issue I'll talk about at the very end. If you look at the longer term goals, as I've said, we need to get better therapies and ways to diagnose the disease early. So, we'll go back to the subtype in a minute. So, if you look at the subtype, it's very easy to determine whether somebody is ER positive, PR positive. It's actually an immunohistochemistry stain that's done in pathology labs. HER2 positive tumors are done the same way. It's a stain. And if you don't get the stain positive with either of those two reagents, the systems that we use, then you're classified as having triple negative. Okay? It turns out that these three subtypes are all found in that large group of adenocarcinomas that are invasive breast cancer. You can't look at an invasive cancer on the histology slide and tell whether or not it's HER2 positive or ER positive. This is two different cancers that are coming out of the same original tissue space. It's the same patient, or different patients, but the same region. So basically, what you've got is a series of cancers. Now, we already know that if you're ER positive, we can get very good responses from anti-estrogen therapy and aromatase inhibitors. And I want to show you how those work in a few minutes, because it impacts on quality of life. HER2 positive tumors have a very good prognosis. Now we're getting to have a very good prognosis. 60 to 70% of these tumors respond very well to a series of drugs called trastuzumab, pertuzumab, or lapatinib. And I swear drug makers make the names as complicated as they can so that nobody can pronounce the drug. It irritates me. This is how cancers work, positive cancers treated. Okay. I want to show you a couple of things. First of all, as I said before, that's the structure of estradiol. Okay? And what I want to point out to you is simply that this is a 4-ring structure. This is the A ring of the structure. And it's got double bonds around it. It's got this fancy structure here. That's called an aromatic ring. Okay? When we talked about aromatase inhibitors, and I'll show you, it's actually, these are named because they prevent this ring from forming, which is critical. So basically the way estradiol works is it binds, it gets it out of the circulation, goes into the cell, it binds to the estrogen receptor. The estrogen receptor binds to the DNA and turns on a lot of different genes in the cell. Those genes are taken out into the cytoplasm, the RNAs, and then it's translated into proteins, and it's proteins that make the cell behave the way it does. And basically, when you've got estradiol present, In the same way it works in a normal, healthy breast, you get cell proliferation. So the growth of the cell is actually controlled, and the growth of the tumor is actually controlled by estrogens. And so you can inhibit it and inhibit the behavior with treatments with something that blocks that pathway. And the one that is commonly used is tamoxifen. Tamoxifen is the most prescribed drug in the world, by far. This is the structure of tamoxifen. And this is now, just to remind you to say that now that it's generically made generically, you don't have to buy the brand name drug. But basically, tamoxifen interferes with the estrogen receptor, binding of estrogen to the estrogen receptor. And what it does is it inhibits the interaction, and so you no longer have this pathway working, and the cells do not proliferate as a result. So what we're doing with all of this is stopping the proliferation. So this works very well in positive tumors where you've got the receptor protein present. It doesn't work in your negative tumors. Okay? You can add as much tamoxifen as you like to a patient who has an ER-negative tumor, and it's not going to stop the cell from growing and dividing. So the cell proliferation in ER-negative tumors is not controlled by estrogens. It's not inhibited by tamoxifen. So the thing is that what you're trying to do, if you've got an ER-positive tumor, what you want to do is to eliminate all of the estrogen present in a woman. Postmenopausal women still have estrogen. They have less estrogen. It's not being made by the ovaries, but it's being made by the adrenal gland predominantly. Okay? And in fact, what the adrenal gland does is it actually synthesizes and secretes what are called adrenal androgens. This is androstenedione. Anybody that's followed baseball for a long time knows that Mark McGwire doped using androstenedione. This is actually the same compound that's involved in the biology of breast cancer. Androstenedione is converted to testosterone very readily. So he didn't dope the testosterone, but he did the next best thing. It's one reaction alone. And the point I want to make to you is that testosterone in the breast and in other peripheral tissues is converted by an enzyme called aromatase. Okay? And what it does, this is not an aromatic ring. Testosterone doesn't bind to the estrogen receptor, but once it's been aromatized, it in fact is produced, and estrogen is produced. And that happens. Aromatase is present normally in the ovaries. The ovaries are no longer functioning. It doesn't do anything, but it can be converted to estradiol in the breast cancer cells. So the reason you add an aromatase inhibitor is because it blocks the adrenal androgens synthesis and binding and conversion to estradiol. So it shuts down and gives you a virtually estrogen-free environment for the individual. Okay. So the drugs that are used, and I'm sure some of you are probably being treated with this, anastrozole, exemestane, and letrozole are actually all being used clinically. And you can actually add letrozole and tamoxifen together, and they are highly effective in preventing recurrence. And a recent study, as in fact the European study, has suggested now that women who are on this and have no recurrence, no breast cancer recurrence, that you can stay on it for as long as you want. It's become a very common feature in Europe. You can stay on this for 10 plus years at least, probably 15. There's some patients in Europe now that have been on it for 20 years. Okay. Are the side effects? I've been told that there are. There's hot flashes. I haven't personally experienced any of these obviously. But there's a lot of irritating side effects with this. Okay. The major one I want to talk to you about is the fact that very low levels of estrogen actually affect the bone health as well. And that's the key feature of these treatments. Very quickly, HER2 treatments. In fact, HER2 is a new form, it's not a new form, it's been around for generations. We finally understand how HER2 actually works. HER2 in a normal cell is present. The actual protein is a receptor that sits on the cell surface of the breast cancer cells. It's also on normal breast cells. And it responds, there's a few copies of the protein, and there's 2 copies of the gene in the nucleus. One of the things about HER2 positive breast cancers is there's actually an amplification of a region of DNA. What this means is just one part of the DNA, instead of having 2 copies, you can have 10, 20, 30, or even 50 copies of that gene. And that gene then puts out a huge number of HER2 receptors on the cell surface. This binds to a protein called ERB2. Okay, it actually binds to the growth factor, but ERB2 is actually the protein. That growth factor stimulates DNA replication and proliferation, and it does so based on the number of copies of HER2 on the surface. So you get a massive increase in the signal to the cell to divide. So the treatments now are using a whole series of proteins that block HER2 action. And they're actually monoclonal antibody-based drugs. Trastuzumab was the very first monoclonal antibody that was ever used in the clinic for any disease. And basically, when HER2 is overexpressed, the cells divide and they will become metastatic. That's part of what their trajectory. But if you add trastuzumab, it actually binds here. There's other compounds that will do the same thing, and they will all block the expression of the activity downstream and turn off the proliferation response. They do one more thing though. They induce the apoptotic response that I talked about earlier on, so the cells die. And the better we get at understanding this mechanism, the more we can add and use combination therapies to actually reduce the recurrence incidence of this disease. Okay. There's also another part of this story that's now emerging. Have any of you heard of Ibrance? Okay. So Ibrance is a really phenomenal, very, very promising new drug that can be used for a lot of different cancers, not just breast. I mentioned to you cells divide. Every time a cell divides, it goes through the cell cycle. Ibrance is one of a very new group of compounds that doesn't affect the estrogen receptor signaling pathway, it doesn't affect HER2 signaling pathways. What it actually does is it stops the cell cycle from working because it inhibits the critical interaction between 2 proteins that regulate the cell cycle. And when these two proteins are inhibited by Ibrance, in fact, what you land up with cells that can't go through the cycle, so they can't divide. Okay? So these actually are being used on postmenopausal women. The major clinical trials have been for ER positive, HER2 negative patients, so the regular ER positive population. The reason for that is entirely due to it's much easier for a drug company to recruit from the 180,000 patients a year that have the disease than a smaller population that is actually more useful. This drug will work very effectively at ER-negative patients. Okay, so it's actually one of the first drugs that will be useful for, very useful for patients with triple-negative breast cancer. And that's the important thing. And I'm not talking today about inflammatory breast cancer, which is a very, very nasty form of breast cancer, because you never diagnose inflammatory breast cancer until it's metastasized already. There's no primary tumor. All you find is metastatic evidence. So it will work for these diseases as well. Okay? So this is going to be a very important new drug. Right now, it's prohibitively expensive. It's $120,000 a year. It's $9,000 minimum for a month. And something has to be done to bring that cost down, obviously. Okay. So, the consequences of these therapies. This is I think my final slide, or my next final slide. I haven't counted, I shouldn't. But the consequences of these therapies, what I want you to understand is everybody in this room has an individual tumor. As scientists, we group you because it's the only way we can do stuff. We haven't got to the stage with breast cancer or most of the solid tumors where we can actually do literal personalized medicine. We're getting there, but it's very difficult to do. But if you look at ER and PR breast cancers, basically estrogen deprivation removes the stimulus to growth. So the most important thing about this is you can have long-term survival with these therapies. But it puts you at risk if you don't do anything for osteoporosis as a major side effect. And there's a very long history. If you look in all of the epidemiology data, there's an increased risk of falls and increased risk of breakage. But basically, we know that vitamin D and calcium supplements can definitely improve bone health. My other half, Dr. Welsh, who's also spoken at To Life, is actually a pioneer in the area of this research. Vitamin D has major effects, good effects on breast cancer. It inhibits breast cancer growth, progression. But it also, with appropriate amounts in the supplement and calcium supplements, also improves bone health. So everybody who's ER positive, whatever the treatment you're on, you should be looking very carefully at bone health. Because what happens with this is, when you run the therapies, every woman who is postmenopausal is at risk for osteoporosis. The risk for osteoporosis goes up on these therapies unless you make sure that you have vitamin D and calcium supplements, and maybe even bisphosphonates as an additional therapy. That's important to do, and that's for every individual to think about. One of the fascinating things that's coming out from studies of HER2-positive breast cancer, and this is work done at Cancer Center as well by Don Compton, who I think is also in here. But basically, if you look at this, HER2-positive breast cancers tend to use lipids as their major source of energy. In other words, they use fats. Okay? So you need to make sure to limit their energy supply, you really do have to have a low-fat diet that deprives tumors of the source of energy that they need for the growth of metastasis. Finally, if you look at triple-negative breast cancers, they tend to use simple sugars, glucose and fructose, as their major sources of energy. And they have an abnormal metabolism. And basically, they use particularly fructose as a very rapid source of energy for growth and metastasis. And so, one of the things that we need to do is to limit the amount of sugars and definitely get rid of any foods that have high fructose corn syrup in them, because that ends up being a much healthier diet from the point of view of looking after the individual and maintaining a very low rate chance of lowering the rate of breast cancer progression. And if you actually look at this, what I'm suggesting is everybody have a healthy diet. That's all I'm really suggesting. And that turns out to be absolutely critical. The same risk factors that were there for breast cancer initiation are actually the same risk factors that are important for progression, and for metastatic progression. So, quick take-home messages, very quickly. It's not one, breast cancer is not one disease. It's many variations on the theme, and I would argue it's as many variations as there are breast cancer patients. It's always going to be individualized. The earlier the cancer is detected, the better the survival rate is. Preventing and treating metastatic disease is really what's probably critical in this process. We've got to stop tumors from getting to the stage where they metastasize. And if they metastasize, the drugs that are being used now, IBRADs among them, is actually very effective. but expensive. Okay? And what we're learning is that molecular subtyping has a major impact on the diagnosis and treatment and survival of breast cancer. And we have excellent therapies, all of this for ER positive and HER2 positive cancers, and maybe less effective ones for triple negative that they're coming through the pipeline. But basically, all of this comes down to understanding the disease a bit more than probably your doctor has told you, but also making sure that you're proactive in your lifestyle. So I'll leave it at that for now. Thank you.